Good morning, everyone. So today I am going to take a class on Clostridium. At the end of this class, you should know different species of Clostridia, the virulence factors, the pathogenesis, clinical manifestations, lab diagnosis, treatment, and preventive measures. So you had a class on Bacillus, no? So Bacillus is a spore-forming aerobic bacteria, while Clostridia is also a spore-forming bacteria, but it is an anaerobic bacteria. So this anaerobic bacteria, they do not have cytochrome system for oxygen metabolism. So they are unable to neutralize the toxic oxygen metabolites. Normally, in those bacteria which can utilize the oxygen for their metabolism, they have certain enzymes, that is a cytochrome system. It contains certain enzymes like catalase, superoxide dismutase, etc. This will help to neutralize the toxic metabolites which are formed as a result of the aerobic metabolism. So the toxic metabolites are usually hydrogen peroxide. Like that, several toxic metabolites will be there, and it will be broken down to water, nascent oxygen. So these enzymes, they neutralize the toxic oxygen metabolites. These enzymes, they are absent or they are present only in smaller amounts in anaerobic bacteria. Accordingly, they are classified into obligate anaerobes and aerotolerant anaerobes. Obligate anaerobe means they can't tolerate even a small amount of oxygen. They can't tolerate oxygen at all. This is because they completely lack the superoxide dismutase and the catalase enzymes. While aerotolerant anaerobes, they possess small amount of the superoxide dismutase enzyme. They completely lack the catalase enzyme, but they possess some other enzyme, the peroxidase enzyme. So because of these enzymes, at least in small amounts, these enzymes are present in these organisms. So they can tolerate oxygen, but they won't utilize oxygen for their metabolism. Okay, so there are two types of anaerobes. One is obligate anaerobes and the other one is aerotolerant anaerobes. For obligate anaerobes, you can remember Clostridium tetani, Clostridium perfringens. All those are obligate anaerobes. For aerotolerant anaerobes, you can remember Clostridium histolytic. Clostridium histolytic is an example of aerotolerant anaerobe. Now coming to Clostridia proper. Clostridia. So this is a gram-positive bacilli having bulging spores. Clostridium is present as a saprophyte in soil, water, in decaying vegetation, animal matter, sewage, etc. So what is the purpose of this organism in this saprophytic, seeing as saprophytes in these conditions is, it will produce a recycling of the organic matter. So it is present in animal matter, decaying vegetation, sewage, etc. It is also seen as a commensal in human intestine, also in other animals. Okay, so the Clostridia, they are motile. They exhibit stately motility, but there are exceptions. Clostridium perfringens, Clostridium tetani, serotype 6. You can remember those two. Sorry for the spelling mistake there. It is Clostridium perfringens, Perfringens. Okay. So the organism, Clostridia, they exhibit stately motility. There are exceptions. One is Clostridium perfringens. Other one is Clostridium tetani, serotype 6. They are non-capsulated except Clostridium perfringens and Clostridium butyrica. So you can remember Clostridium perfringens for both. That is, all Clostridia are motile and non-capsulated except Clostridium perfringens, which is non-motile and capsulated. Okay, so you can remember like that. Now it has spores. So the spore formation occurs in unfavorable condition. Almost all the Clostridia, they produce subterminal spores. Subterminal spores. I will show you the picture, but there are exceptions. The exceptions are Clostridium tetani, it will produce terminal spore, which is spherical. This gives a drumstick appearance. Then Clostridium tertium, it produces terminal spores, which is oval. So this gives a tennis racket appearance. And Clostridium bifermentans, it produces central spores, which are oval. So these are exceptions. So you can see this is the subterminal spore. Most of the Clostridia, they produce subterminal spores only, but the exceptions are Clostridium tetani that produce terminal round spores, spherical spores, which gives a drumstick appearance. Then Clostridium tertium, it produces terminal oval spores, which gives a tennis racket appearance. Then Clostridium bifermentans, which produce oval and central spores. That is about the spores in Clostridia. Now coming to the cultivation. You had classes on systematic bacteriology. You will be knowing that we can cultivate this organism in Robertson's cooked meat broth. That is a anaerobic culture medium or thioglycolate broth. These can be used for the cultivation of Clostridia. This Robertson cooked meat broth, it contains minced cooked meat particles from the beef heart. So this will contain glutathione and unsaturated fatty acids. Both these are substances which take up oxygen and create a lower redox potential, thus maintaining the anaerobiosis. Okay, so this is how RCM is maintained as an anaerobic medium. So in this media, if there is growth, it will appear as a turbidity, and there will be changes in the meat particles. If the organism is a proteolytic organism, the meat will turn black and there will be a foul odor. Examples are Clostridium tetani, Clostridium botulinum A, B, and F, serotypes. Okay, and if the organism is a saccharolytic organism, it will turn the meat pink. Example is Clostridium perfringens, difficile, and Clostridium botulinum serotype C, D, and E. Okay. So in this picture, you can see the extreme left is uninoculated media. It contains the minced meat particles and the broth above. It is not turbid, it is uninoculated. So these two are inoculated. You can see there is a change in color of the meat particles. Here it is pink, so it is a saccharolytic organism. Also the medium has turned turbid, so there is growth, indicating growth. And the meat has turned black here, so it is a proteolytic organism, and there is turbidity in the media, indicating that there is growth. Now coming to Clostridium perfringens. In this class, I will be taking only Clostridium perfringens. The remaining, the Clostridium tetani, difficile, we will deal with in the next class. Okay, so Clostridium perfringens, it was previously known as Clostridium welshi. It is a common cell in the large intestine of human beings as well as other animals. It is capsulated, it is non-motile, and a gram-positive bacilli, which produce subterminal bulging spores. Okay, so this organism, it produces several toxins and it produces invasive infection. It produces invasive infection as well as it is toxigenic. It produces 4 major toxins, which are termed as alpha, beta, epsilon, and iota toxin, and 8 minor toxins, gamma, delta, lambda, kappa, theta, eta, mu, and nu. These are the minor toxins. So 4 major toxins and 8 minor toxins. And several other substances such as neuraminidase, histamine, bursting factor, circulating factor, all these are produced by Clostridium perfringens. Also, it produces an enterotoxin, which is responsible for the food poisoning and other gastrointestinal infections. Now coming to the clinical features. Clostridium perfringens produce a spectrum of infection. It can produce wound infection, enteric infection, and it can produce other infections like bacteremia, then brain abscess. All these can be caused by Clostridium perfringens. So coming to wound infection, wound infection, it can be classified into simple wound infection, anaerobic cellulitis, and anaerobic myositis. Anaerobic myositis is also known as gas gangrene. Okay, simple wound infection means there is infection of the superficial area of the wound only. There is no toxin production. Okay, so that is simple wound contamination. And anaerobic cellulitis means there is invasion into the facial plane. Then it is known as anaerobic cellulitis, and minor amount of toxins are also produced. Then third one is anaerobic myositis. Here, the organism invade into the muscle and produce large amounts of toxin, and there will be gas inside the muscle, giving the crepitus inside the muscle. So these are the types of wound infection. This wound infection, it occurs because of the two favorable conditions that occur during wound two favorable conditions that promote the growth of the Clostridium perfringens. That is, when there is trauma and all, there will be entry of Clostridium perfringens spores which are present in the soil and all. In case of road traffic accidents like that, there will be entry of spores present in the soil into the open wound. That is one factor. And the second factor is during trauma, there will be crush injuries, no? So like in road traffic accidents, there will be crush injuries and all. So the blood supply to the area won't be proper. So this will produce a anoxic state in the tissue. That is a lack of oxygen in the tissue. This will create a favorable environment for the growth of Clostridium perfringens. So these two factors promote the infection, that is a growth of Clostridium perfringens in the wound, causing wound infection. Okay, so now coming to the enteric infection. This is due to Clostridium perfringens type A enterotoxin, and this occurs during consumption of improperly cooked meat containing the Clostridium perfringens. And we can diagnose the infection by detection of this toxin in the feces by enzyme immunoassay. The second type of enteric infection is, this one is the first one, the enteric infection caused by type A enterotoxin is food poisoning. Food poisoning caused due to consumption of improperly cooked contaminated meat. Okay. Other enteric infections caused by the organism is enteritic necroticans, which is also known as gas gangrene of the bowel. It is a fatal illness, like it manifests as a necrotic lesions in jejunum, and it is also known as pig bell in other languages. In other languages, it is known as pig bell. It is a fatal condition. There is necrosis of the jejunum. Okay, and next one is necrotizing enterocolitis, and it can also cause gangrenous appendicitis. So it can cause food poisoning, enteritis necroticans, necrotizing enterocolitis, and gangrenous appendicitis. So these are the enteric infections caused by Clostridium perfringens. And it can also cause other infections such as bacteremia, skin and soft tissue infections, and infections of the endometrium, then meningitis, brain abscess, panophthalmitis, etc. Now coming to the most important part, that is the gas gangrene. So gas gangrene, it can be defined as a rapidly spreading edematous myonecrosis. It occurs in association with severely crushed wounds. I have told you earlier, no, there are two factors that promote the growth of organism in the areas of crushed wounds. So because of those two factors, there will be rapidly spreading edematous myonecrosis in association with the severely crushed wounds. This is also known as malignant edema or Clostridial myonecrosis. And several agents are said as established agents for gas gangrene. First one is a Clostridium perfringens only, that is followed by Clostridium novi and Clostridium septica. one thing you have to remember, the gas gangrene produced by Clostridium perfringens will be associated with infection by other Clostridia species. So it is polymicrobial. Mostly the infections, all the infections caused by the Clostridium perfringens is polymicrobial. Gas gangrene is typically polymicrobial only. So along with the Clostridium perfringens, you will see other species of Clostridium also with it. So the established agents are Clostridium perfringens, followed by Clostridium novi and septicum. Then there are probable agents which causes rarely. They are Clostridium histolyticum, Clostridium sporogenes, Clostridium phalanx, bifermentans, and sodali. So these agents are also considered as probable agents of gas gangrene. The pathogenesis I have described already, the anaerobic environment and contamination of wound with Clostridium spores, and followed by the virulence factors that are produced by the Clostridium perfringens, leading to the gas gangrene. That is, organism will proliferate locally. So it produces several toxins. No, I have said earlier. So the mainly the two toxins, the alpha and the theta toxin. And the alpha toxin, it acts as a phospholipase and a sphingomyelinase. So it has two activities, phospholipase activity and sphingomyelinase activity. It activates the platelet adhesion molecule GP2B3A and neutrophil receptors CD11B, CD18, and this result in aggregation of platelets and neutrophils leading to vascular occlusion. Okay, this is the pathogenesis. And also it suppresses the myocardial contractility, which leads to reduced cardiac output and hypotension. So this is the picture of gas gangrene. Actually, here you can see gas present in the muscle, in the muscle layer. And these are the necrotic regions. So the clinical manifestations, they vary, the incubation period, it vary based on the nature of the injury, wound contamination, and the type of species. So for Clostridium perfringens, the incubation period is 10 to 48 hours. It is less compared with other species. For other species, for Clostridium septicum, it is 2 to 3 days. And for Clostridium novi, it is 5 to 6 days. And the clinical features are, there will be sudden onset of very severe pain at the region affected. And there will be a rapid foul smelling, thin, serosanguinous discharge, that is blood-stained thin discharge, serous discharge mixed with the blood. It will be foul smelling also. And because of this gas getting collected in the muscle, you can hear crepitus over the muscle, and there will be edema. And finally, the tissues, they get devitalized and they will liquefy and they will slough off. So the gangrene, it will slough off. And later on, the patient can go to shock, organ failure, and even death can also occur. Coming to the laboratory diagnosis of Clostridium perfringens. So the laboratory diagnosis, it has a role only for confirmation and species identification. That is, you shouldn't wait for the laboratory report to arrive. You should start the treatment immediately. Upon clinical suspicion itself, you have to start the treatment. Okay, so the specimens which are processed are the necrotic tissue, the muscle fragments, and the exudates, which are deep exudates. You should collect from the depth of the wound. There only anaerobiosis is there, and there only we can isolate the organism from there only. So necrotic tissue, muscle fragments, exudates from the depth of the wound, and blood. These are the specimens that are usually collected. And the specimen, it should be put in Robertson cooked meat medium, and it should be transported immediately to the laboratory. Okay, so in the laboratory, the first thing that we do is always gram stain only, and you will see the gram-positive bacilli, and the absence of neutrophils, it is a typical finding. Absence of neutrophils is a typical finding. Regarding the presence of spores, usually Clostridium perfringens, we can't see the spore in the clinical sample. Clostridium perfringens, we can't see the spore in the clinical sample. For other species, I have told now it is polymicrobial only. And for other species, spores may be seen that I will describe later. Okay. So the absence of neutrophils is a peculiar feature. And we should incubate the plates anaerobically at 37 degrees Celsius for 2 days. That is, after inoculating onto the media, in anaerobically, we should incubate at 37 degrees Celsius for 2 days. And for identification of the organism, there are some peculiar features. One is the target hemolysis that we see on the blood agar. For Clostridium perfringens, we see target hemolysis on the blood agar. This is the picture of target hemolysis. Actually, target hemolysis means there are two zones of hemolysis. There is a double zone of hemolysis. You can see there is a hemolysis, a zone of hemolysis which is complete zone of hemolysis within, and a incomplete zone of hemolysis outside. The inner complete hemolysis is due to the theta toxin, and the outer incomplete hemolysis is due to the alpha toxin. This is a feature of Clostridium perfringens. The second one is the Nagler reaction. That is, when we inoculate the organism on the egg yolk agar, you can see opalescence surrounding the colonies. This is the area where the organism was inoculated, and you can see in this portion, you can see there is opalescence surrounding the colonies. This is because of the lecithinase activity of alpha toxin. You can use either egg yolk agar or media containing 20% human serum. Okay, in both these media, it will show the same feature only, opalescence. And you can confirm it by adding the anti-alpha toxin. When you add the anti-alpha toxin, what happens? The alpha toxin is neutralized now. So there is no lecithinase activity. The opalescence has gone. So this is known as Nagler reaction, which is shown by Clostridium perfringens. And the third feature that you see is the reverse Camp test. It is positive. That is, on blood agar, we inoculate Clostridium perfringens at the center, and perpendicular to that, we inoculate Streptococcus agalactiae, group B Streptococcus agalactiae. So there is a enhanced zone of hemolysis, looking like an arrow pointing towards the Clostridium perfringens. This is known as reverse Camp test. This is positive for Clostridium perfringens. The other features are, it has a property of heat tolerance. So even at 45 degrees Celsius, it can grow in the RCM broth for 4 to 6 hours. So this is a property of Clostridium perfringens. And in litmus milk, it can ferment the lactose. So on fermentation, there will be production of acid and gas. So this will give a stormy clot-like appearance. Stormy clot reaction is seen in the litmus milk. And other biochemical tests are performed to identify the organism. So all these are done to identify the species and to confirm the diagnosis, but we shouldn't wait for the laboratory report. Immediately on clinical suspicion, we have to start treatment for Clostridium perfringens. Okay, coming to the treatment, the most important thing is early surgical debridement and a wide zone of clearance. That is, we should remove all the devitalized tissue. All the devitalized tissue should be removed completely, and we should delay the wound closure for 4 to 6 days. Only after all the infection settles down, we should close the wound. So early surgical debridement, and also it should be completely, the devitalized area should be completely debrided, and the wound closure should be delayed for 4 to 6 days. And the antibiotics, penicillin and clindamycin, to be given for 10 to 14 days. And we can give hyperbaric oxygen therapy because this organism won't tolerate oxygen now. So we can give hyperbaric oxygen. This will destroy the organism. And we can give passive immunization with anti-alpha toxin also. This also has some role in treatment. And regarding the prevention, the vaccines against alpha toxin, they are under trial only for human beings. There are studies in animals, but they are only under trial for human beings. So that is all about Clostridium perfringens. The next class will be on Clostridium tetani, difficile and all. So in order to summarize, Clostridia, most of the organisms, they are motile and non-capsulated, except the Clostridium perfringens, which are non-motile and capsulated. They are gram-positive bacilli having bulging spores, and they are seen as saprophytes in the environment and also as a common cell in the intestine of vertebrates and invertebrates. And regarding the spore, almost all produce subterminal spores, and you know the exceptions. The drumstick appearance for Clostridium tetani, for Clostridium tertium, the tennis racket appearance, and for bifurcates, it produces central oval spores. Regarding the cultivation, the RCM and some proteolytic organisms, they produce meat black, saccharolytic produce meat pink. And about the perfringens, the 4 major toxins, 8 minor toxins, and the spectrum of diseases that they produce: wound infection, enteric infections like food poisoning, enteritis necroticans, necrotizing enterocolitis, gangrenous appendicitis, and other infections like bacteremia, skin and soft tissue infections, all those infections. And about the gas gangrene, and it is a rapidly spreading myonecrosis associated with the crushed wounds. And most established agents are Clostridium perfringens, followed by novi and septicum. And the virulence factor, the main action is by the alpha toxin. It is alpha toxin that produce almost all the features that will lead on to the disease. Among the clinical manifestations, sudden onset of pain, there is crepitus in the muscle, leading to edema, gangrene. And regarding the laboratory diagnosis, the gram stain, there is absence of neutrophils and target hemolysis on blood agar, Nigler reaction, reverse cam test, and in litmus milk, stomy clot reaction. So regarding treatment, the most important thing is early surgical debridement and antibiotics, penicillin and clindamycin. Okay, that is all. Thank you.